Hi there, and welcome to a video outlining the electron transport chain. My name is Corey Andres. I'm from Central New Mexico Community College using our CNM glass system. So for the electron transport chain in prokaryotes, which is what I'm focusing on here, I'm just going to kind of orient us as to where we are. So I'm going to call this the cell wall because I'm not including the membrane just yet. I'm drawing a thick peptidoglycan layer, which should kind of trigger in your mind that this is a gram positive, but you would see the same process in gram negatives as well. So in the cell membrane, of course, just to remind you, that's your phospholipid bilayer. You have various proteins that are embedded in the membrane that can assist in this process. And it varies in prokaryotes. In a eukaryotic system, we could name them all and we could say, okay, it's this one first, this one, that. The number and type will vary here. So I'm just going to kind of put a number in here. I'm not going to name a lot of them. I will mention that a lot of these are cytochromes, and there's different types, but that's not the only type. What those do is when you have your electron carriers arrive, because everybody wants to know, why do we have to learn about all these electron carriers? NADH and FADH2. And again, that number may vary as well. If we're talking about a aerobic system, 2 is a common number and 10 that are arriving at the electron transport chain. But in an anaerobic respiratory system, it may vary. So one of them may even be absent. Okay, so those arrive at the chain. And so on those molecules, you have this H part. And that's going to move through the chain. And as it does, each one of these the transport proteins is successively more what's called electronegative than the other, meaning that it can pull off electrons from its neighbor, basically. It's an electron hog. Sometimes people use that type of terminology. So it pulls it off from the preceding one. And as that happens, it's split up such that you've got electrons building up on this side, and then the H, right, a hydrogen, has one proton and 1 electron. So the protons, so you often see an H+, are building up out in the space. And it's nice that you have the cell wall, because otherwise this is just the environment that you have out here, and they would just kind of escape. So, but it can affect the efficiency of the system a little bit more so where eukaryotes have a mitochondria. Everything is nicely contained in the organelle. You don't have all this sort of leakage of protons. However, here you can have some leakage, so you can lose efficiency. So when we give numbers for ATP production, those are ideals. They do vary a bit. And also varies a bit on proteins and whether we're in an aerobic or anaerobic electron transport chain. So this is our ATP synthase. And I always think of this as the car battery situation. You've got your positive on one side and your negative on the other, because I think a lot of people can relate to the two cells. They've jumped the car battery. They know that there's a separation. When you open up that connection, these H pluses can leak over, and sometimes these are just shown as pluses. And that's what provides the energy. Basically, it hits the electrons and there's an energetic reaction there. And that's what provides the energy to take inorganic phosphate, okay, which are getting recycled from other ATPs that were used up. You're going to add that to ADP. Well, gosh, why can't we just do that one step? That would be nice. But we need to go through all the successive steps of breaking down nutrients, substrates, going through the various stages of pulling off the electrons very slowly, being carried over by these electron carriers, and then finally being brought over to this electron transport chain to provide energy. So this whole process, all the steps that you have to go through, that you probably feel like you're personally going through, that outline, glycolysis and then Krebs and then getting here, is all about this last part. Having enough energy to get the phosphate group joined to the ADP. So, a lot of times when people are looking at things like glycolysis and Krebs, they're really focusing on the ATP that's made there. That's kind of small potatoes in some ways. I mean, if some people or some organisms only have a fermentation pathway, that's a big deal for them. But really the primary production of ATP is going to be down here. And this makes, you'll see in some anaerobic pathways can be pretty low. And I cannot spell anaerobic today. Let's try that one more time. So anaerobic pathways can be at that lower end, but they can be higher as well. But the highest one most likely is going to be your aerobic respiration in most cases. So that's really, in many ways, your ideal in terms of making ATP. So why have any organisms evolved to be anaerobic if they're at that lower end? Well, keep in mind if you're anaerobic, there's very few of competing species as opposed to an aerobic environment where you'd have thousands, millions of competing species. So you may not get as much ATP, but you might have first grab at the substrate. So if it's, whether it's glucose or whatever the starting substrate is. So just to reminder here, in aerobic pathways, so the aerobic electron transport chain, your final electron acceptor, so here's our successive electron acceptors, but our final electron acceptor, which is going to be over here somewhere, this will be our final electron acceptor, using shorthand for electron. Is O2. And that will make H2O as the waste, okay, when the electrons are joined during that, reduction step. In anaerobic, ETC, your final electron acceptors can vary. So it might be something like, and forgive me here, I'm not going to have all my pluses and minuses probably sorted out, but something like your sulfates, carbonate, nitrates, and they will make end products or waste products like H2S, or sometimes you'll see that written as HS. Okay, so you see I'm adding the hydrogens on here, and during the, because I'm doing a reduction reaction. H2S, methane, ammonia, even we'll see it totally reduced. So you'll have a very different set of products. So you want to look for the water and the oxygen to indicate that it's aerobic, and the anaerobic, anything else goes, anything that's non-oxygen that's there. Yes, oxygen is present, but it's not elemental, it's not free oxygen, the O2, that's different than just having oxygen in the compound, because it's not free in quite the same way. Okay, that's what I have for the electron transport chain. I thank you very much, and please contact